Plasmids and episomes. Plasmids and episomes are the extra chromosomal genetic elements that play a crucial role in bacterial genetics, adaptation, and survival. Plasmids and episomes are extra chromosomal genetic elements that play a crucial role in bacterial genetics, adaptation, and survival. Definition and basic characteristic features here. Structure: plasmids are autonomously self-replicating double-stranded DNA molecule that exist in a free state within the cytoplasm of a cell. While they are mostly circular, cyclic in shape, exceptions exist, such as in Borrelia, which possess a linear plasmid. In Borrelia, plasmids are linear, but excepting them, while mostly circular, plasmids are autonomously self-replicating double-stranded DNA molecule that exist in a free state within the cytoplasm of a cell. While they are mostly circular, cyclic in shape, exceptions exist such as in Borrelia, which possess linear plasmids. Episomes. Episomes, when a plasmid has the ability to reversibly integrate into the bacterial chromosomal DNA, it is specifically referred to as an episome. When a plasmid has the ability to reversibly integrate into the bacterial chromosomal DNA, it is specifically referred to as an episome. Once integrated, the episome behaves like a segment of the host chromosome and replicates synchronously with it. Once integrated, the episome behaves like a segment of the host chromosome and replicates synchronously with it. Size and quantity. Size and quantity. They represent only a small fraction of the bacterial genome, typically ranging from less than 1% to about 10% of the size of the bacterial chromosome, around 300 genes compared to the 3000 genes of a bacterial chromosome. A single bacterium can contain anywhere from just 1 to more than 40 plasmids. Essentiality. Plasmids are not essential for the normal physiological functioning, life, or reproduction of the host bacterium. Because they are not strictly necessary, bacteria can gain or lose plasmid during their lifetime. The process of naturally or artificially eliminating a plasmid from a bacterium is known as curing. Essentiality: plasmids are not essential for the normal physiological functioning, life, or reproduction of the host bacterium because they are not strictly necessary. Bacteria can gain or lose plasmid during their lifetime. The process of naturally or artificially eliminating a plasmid from a bacterium is known as curing. Functions and properties conferred by plasmid. Although they are not essential for basic survival, plasmid frequently carry genes that confer a selective survival advantage to the bacterium in specific environment. Some plasmid do not outwardly express their traits and are known as cryptic plasmid. When expressed, plasmids can provide the following functions like antibiotic resistance. The plasmids carry genes that make bacteria resistant to various antibiotics like penicillin, tetracycline, erythromycin, chloramphenicol. This resistance is frequently due to plasmid-coded enzymes like beta lactamases, like penicillinases that destroy the drugs. Virulent factors. Plasmid often code for toxins, adhesins, and other elements that enhance a pathogen's virulence. Examples include the heat labile or heat stable enterotoxins in Escherichia coli, the capsule gene PXO2 in Bacillus anthracis, the epidermolytic toxin B in Staphylococcus aureus, and the bundle forming pili used by some E. coli for attachment. Bacteriocin production. The col plasmids contain genes that code for bacteriocin, such as colicins in coliform bacteria or staphylococcus in Staphylococcus aureus, antibiotic-like substances that selectively kill or inhibit competing bacteria. Metabolic and environmental adaptations of the plasmids. Metabolic plasmids enable hosts to degrade complex organic molecules, utilize various sugars and hydrocarbons, and fix nitrogen. They also provide resistance to toxic heavy materials like mercury and silver and produce light resistant DNA repair enzymes. Classification of plasmid. Plasmids are primarily classified based on the following criteria: fertility of plasmid fertility, code for sex pili needed to form conjugation tube; resistance plasmids, code for antibiotic resistance; coal plasmid, code for bacteriosin; virulence plasmid, code for toxins and cellular adhesin; metabolic plasmid, facilitate distinct metabolic activities. Based on the transferability, conjugative self-transmissible conjugative or self-transmissible plasmids, these possess the genetic information transfer factor required to initiate their own transfer from a donor cell to a recipient cell via a conjugation tube. Non-conjugative or non-self-transmissible plasmids. These lack the transfer apparatus and cannot transfer themselves independently. However, they can still be transferred if assisted by a transfer factor or a bacteriophage. Based on compatibility, based on compatibility, closely related plasmids are incompatible and cannot stably coexist inside the same bacterial cell. Unrelated plasmids can coexist stably and are said to belong to the different compatibility group. Role of plasmids in gene transfer. Plasmids are the key drivers of horizontal gene transfer among bacteria. Conjugation. This is a mating process where a donor cell containing an F plasmid forms a physical connection, sex plus, with a recipient cell lacking the plasmid and transfers a copy of the plasmid. If the F plasmid is integrated into the bacterial chromosome and episome, it creates high frequency recombination strain capable of transferring large segments of the host's chromosomal DNA. Transduction. In some instances, such as the penicillinase plasmid in staphylococci, plasmid DNA can be picked up by a bacteriophage, a virus that infects bacteria, and transferred to another bacterium. Practical adaptations of plasmids. Genetic engineering. Due to their ability to autonomously replicate and transfer DNA between cells, plasmids are widely used as a cloning vectors. Scientists can artificially insert foreign genes into a plasmid and introduce it into a bacterial host to produce large quantities of therapeutic proteins like human insulin, growth hormone, or hepatitis B vaccine. Environmental cleanup. Because metabolic plasmids can degrade complex and unusual compounds, they are being researched for use in detoxifying environmental wastes and pollutants. They're all about plasmids.